What would it take to help a completely severed tendon reconnect? That question sits behind one of the most striking studies in the BPC-157 story. And it is a major reason this experimental peptide has generated so much interest. Before we discuss what that research may mean for humans, let's look at what the researchers actually did. I'm Dr. Kish Carlton with Mind, Body, Spirit Medicine. In this episode, we're taking a closer look at BPC-157, why its healing potential has attracted so much attention, what the research actually shows, and what you should understand before considering it. Let's dive in. In a 2003 rat study, researchers completely transected the Achilles tendon, creating a substantial gap between the cut ends. A complete transection creates a much more demanding repair problem than ordinary overuse or a mild tendon strain. This was not a minor strain. It was a dramatic injury model designed to test whether repair could be meaningfully improved. Compared with controls, the treated rats demonstrated better functional recovery and stronger mechanical performance in the experimental model. Functional recovery in this context means the animals regained useful movement more effectively than the comparison group. The reported differences included greater load to failure and improved elasticity measurements, findings that naturally drew attention. Researchers also described improved fibroblast and collagen organization, a smaller tendon defect, and eventual restoration of tendon integrity in that model. Mechanical testing asks a different question. How much force can the repaired tissue tolerate, and how does it behave under stretch? That is a remarkable preclinical finding, but it is not proof that BPC-157 regenerates severed human tendons. The excitement comes from the combination of a severe injury, measurable recovery, and a biological pattern that appears to support tissue repair. The eventual restoration of tendon integrity is precisely the kind of observation that makes researchers want to investigate further. For scientists, that creates an opportunity. Understand the signal, test it carefully, and find out whether it can translate. BPC-157 is commonly described as a gastric pentadicapeptide, meaning a peptide associated with a 15 amino acid sequence. The scientific opportunity is real, but the word regeneration should be used carefully when the evidence comes from rats. Its gastric context helps explain the original interest, but natural presence does not automatically establish safety or clinical effectiveness. In tendon research, the central question is whether the signal can support organized repair rather than simply produce a temporary change in symptoms. A healing tissue is an environment, not just a strand of collagen, and several systems must work together. One proposed mechanism involves fibroblast migration, bringing repair-oriented cells toward damaged tissue. Another involves collagen organization, because the quality and alignment of new tissue may matter as much as the amount produced. Better organization may matter because aligned collagen can influence strength, elasticity, and how tissue handles load. Angiogenesis, or the formation of new blood vessels, is also proposed as a way to support the repair environment. BPC-157 has also been studied in relation to nitric oxide signaling, which can influence blood flow and inflammatory regulation. These mechanisms may not be isolated switches. They may form a network that changes the repair environment in several ways. Researchers have explored possible effects involving growth hormone receptor expression, another intriguing pathway that still needs careful translation. Inflammatory regulation may be part of the picture, but reducing inflammation is not the same as rebuilding mature functional tissue. Mechanisms can be scientifically compelling while the actual patient outcome remains an important unanswered question. Tendon-to-bone healing is especially complex because the attachment zone must transition between different tissue types. Other preclinical work has examined tendon-to-bone healing, another area where structure, attachment, and mechanical function all matter. Muscle injury models have also contributed to interest by raising questions about tissue repair and recovery after damage. In muscle models, researchers are asking whether the signal influences repair biology without confusing repair with guaranteed performance recovery. The gastrointestinal findings are especially relevant to the peptide's gastric context, including preclinical observations involving mucosal protection. Wound repair models add another layer, suggesting that the research may involve several connected aspects of tissue biology. For gastrointestinal research, the interesting question is whether barrier protection and tissue repair can be separated from broader disease claims. That breadth is why scientists and clinicians find BPC-157 intriguing. The signal appears across different repair settings. This is not one isolated claim. It is a cluster of preclinical observations that point toward a possible regenerative biology. Wound models are useful because they let researchers observe closure and remodeling, but they still do not reproduce every human wound. The challenge is that an encouraging signal must pass through many translation steps before it becomes a reliable clinical therapy. Cell studies can reveal pathways and responses, but they cannot reproduce circulation, immune complexity, movement, or recovery in a person. A promising signal becomes more persuasive when different teams can reproduce it under clearly described conditions. Animal studies add important biological context while still leaving questions about species differences in absorption, metabolism, and response. The human evidence is small, but the fairest description is limited rather than non-existent. Human evidence can begin with observations, but each study design answers a different question and carries different uncertainty. Early human observations can suggest that a question deserves more study without settling whether the treatment works. Small uncontrolled reports cannot reliably separate treatment effects from natural recovery, expectation, selection, or other care. A strong trial would need meaningful endpoints such as function, strength, healing, quality of life, and adverse effects. Large, well-designed controlled trials are the next chapter because they can test benefit, harms, and meaningful outcomes more reliably. The lack of large controlled human trials does not prove the peptide cannot work. It means the key clinical question remains open. Efficacy asks whether something helps. Safety asks what it costs, who is at risk, and what happens over time. For anyone considering treatment, product identity is a separate question from biological promise. There is no FDA-approved BPC-157 product, no validated clinical formulation, and no established standardized dose to rely on. That matters because an approved medicine comes with a defined review and quality framework that experimental products do not share. Even a scientifically interesting molecule cannot answer questions about a product whose identity or quality is uncertain. Current regulatory concerns include impurities, active ingredient characterization, immunogenicity, and limited human safety information. Compounded, imported online, and research use only products can raise additional questions about sourcing and quality control. The point is not to dismiss the science. It is to match the confidence of the claim to the quality of the evidence. Tested athletes should verify current anti-doping rules with the governing bodies that apply to their sport and event. If treatment is being considered, supervision should mean informed discussion, monitoring, and a clear plan for reassessment. If you are considering treatment, begin with the diagnosis and the problem you are actually trying to solve. Ask what established alternatives exist, what evidence supports them, and why they may or may not fit your situation. A supervised conversation should also define monitoring, reassessment, possible adverse effects, and stopping criteria. Those questions help turn enthusiasm into a more informed and clinically responsible discussion. The most useful conversation is neither hype nor dismissal. It is an honest match between expectations and evidence. BPC-157 may represent a genuinely interesting regenerative signal, and the animal findings deserve serious scientific attention. At the same time, remarkable animal findings are not the same thing as proven human effectiveness. We should be curious enough to study the opportunity and disciplined enough not to overstate what we know. The human clinical evidence has not yet caught up with the remarkable findings seen in animals. That balance leaves room for discovery while protecting patients from conclusions the evidence cannot yet support. This video is general education, not individualized medical advice, and it does not establish a physician-patient relationship. Treatment decisions should be made with a qualified clinician who knows your medical history. If you found this helpful, subscribe to the channel, explore the complete peptide therapy series, and visit Mind, Body, Spirit Medicine to learn more. I'll see you in the next episode.